The genetic condition that we will be overviewing for our group final project is Angelman Syndrome, so thank you for taking the time to listen.
Hi, this is Alexis talking, and I will be going over what Angelman Syndrome is and how it would be explained by as a nurse. Angelman Syndrome is a rare neurological and genetic disorder. It is caused by a loss of function in the UBE3A gene in the 15th chromosome coming from the mother. In most humans, the UBE3A gene from the father is silent, meaning it does nothing in children. Our UBE3A gene from our mother helps the brain develop. However, with Angelman syndrome, the gene from the mother is problematic, causing problems with neurological aspects such as speech and movement. Symptoms of Angelman syndrome include walking and balance issues, GI concerns, seizures, and difficulty with speech. Developmental delays begin around birth all the way up to the age of 24 months. Seizures begin between 18 months and 3 years of age is when these seizures usually start to develop in Angelman syndrome. Symptoms are similar to autism and cerebral palsy, and therefore Angelman syndrome is usually misdiagnosed, and it is uncertain of what actually causes it.
Hello everyone, this is Kayla speaking. My portion will be touching on Angelman syndrome patterns of inheritance and family history. It is noted that this genetic condition is rare, and according to Mayo Clinic, researchers usually don't know what causes the genetic changes that result in Angelman syndrome. Interestingly, most people with Angelman syndrome don't have a family history of the disease. So without having a child presenting with physical and behavioral implications of Angelman syndrome, family members are unlikely aware of the genetic mutation that they could possibly be carrying. The statement helped me to understand that determining a potential family history of this genetic condition is rather difficult, and laboratory diagnostic testing is required for the diagnosis. So as a nurse, my first task would be questioning the parents regarding developmental delays and other abnormal physical findings present in their child. So I would ask questions about their movement. Are they clumsy, unsteady? Are you witnessing quick and unusual jerky movements? One example would be the uplifted hand flapping movement. Are the parents noticing excessive laughter and smiling? Do they have an easily excitable personality? I would also discuss their speech patterns. Is there an impairment? Are they babbling yet, using minimal words, or are they nonverbal, because that can flag a concern? And also I would ask if there have been any incidents of seizures. My second task would be to confirm or rule out the diagnosis of the affected child. So after the discussion with the parents about the developmental and physical findings, if Angelman syndrome is indeed suspected, I would encourage a blood test for DNA methylation in order for a diagnosis to be confirmed, and I will go into further detail on the next slide. The two figures I have shown here better explain the genetic mechanisms that are actually taking place to cause Angelman syndrome. The first chart on the left, I wanted to focus on just the first four mechanisms that are listed. To start, majority of individuals have acquired Angelman syndrome through deletion, meaning roughly 70% of the time this genetic condition is acquired through non-inherited spontaneous mutation of the chromosome. And then uniparental disomy is another spontaneous non-inherited mechanism. Imprinting defect is one of the inherited mechanisms, only accounting for 3 to 5% of Angelman syndrome cases. And then the UBE3A mutations can either occur spontaneously or be maternally inherited, and this accounts for 5 to 10% of Angelman syndrome cases. So as you can see, inherited mutations rarely occur. Now, the figure on your right is a flowchart that goes through the different lab and diagnostic tests that determine which of the genetic mechanisms that cause the individual's Angelman syndrome. The flowchart begins with a blood test for DNA methylation, and if that test result is normal, they will do the UBE3A gene testing, and if that comes back positive, the individual received Angelman syndrome through the UBE3A mutation. And then if the DNA methylation test result was abnormal, 3 mechanisms are possible, which is the chromosome deletion, the UPD, or imprinting defect. And in order to figure out those, they do a FISH or CGH study. And if that either of those come back positive, chromosome deletion was the cause of Angelman syndrome, which is the 70% occurrence. And then if the FISH or CGH study comes back normal, they will do a DNA marker analysis, and if that is positive, UPD was the cause for Angelman syndrome. And then if that DNA marker analysis is normal, imprinting defect is presumed, since a chromosome deletion and UPD have been excluded.
The specific role of RN when it comes to Angelman syndrome is really recognizing early detection of this and different symptoms such as attacks of gait, continuous happiness, smiling, flailing of the arms, things of that nature. A lot of these kids are diagnosed 6 to 12 months, and majority are misdiagnosed. It is a rare syndrome. It's a teaching thing as well. You know, you really want to teach the parents about the treatments that are possible, which is basically supportive of other underlying health issues that this causes, such as seizures and different communication problems, things like that. And really, it's all about getting them in touch with the right resources, because this is a lifelong thing that their child will have, and providing them with resources such as Angelman Syndrome Foundation to really help them and get them in with support from other people going through the same thing. Recommendations and referrals, we would want to refer them to physical therapy. A lot of these kids can live pretty physical, active lives and hold jobs and everything like that, but the earlier you get physical therapy involved, the better off they will be with, gait and movement. They may need possible surgical, but with good physical therapy, they should be well off. They may need to be referred to an ophthalmologist for possible strabismus, and also behavioral therapy. Again, it's just the same as physical therapy. The earlier you can start things like that to help them get more acclimated and normal, the better off you are. Same with communication therapists. It can help them to develop better speech. And also a genetic counselor for possible future children and DNA testing in that regard. Just letting the parents know that that's available. The family discussions, you really want to talk about, the possible seizures that could happen at 2 to 3 years of age, when those could start. You want to talk about some of the abnormalities and symptoms. So no crawling or babbling, at 12 months of age. The intellectual disabilities, no speech or minimal speech, difficulty walking, moving or balancing well, frequent smiling and laughter, happy, excitable personality, sleeping problems, possible future things such as scoliosis. And you would want to talk about their options as far as seizure medications and things such, and then supportive resources and also future expectations. So let them know that people with Angelman syndrome can lead very good lives and have jobs and go to school. Informatics aspects of Angelman syndrome is really about using informatics and computers and such to monitor their health over the span of their lifetime and to help treat underlying medical issues. You can do that with MRIs, EEGs, different diagnostic testing, help with seizures and other medical issues that arise from having Angelman syndrome. Also, information handouts, different pamphlets and stuff to really educate the public and the parents on the syndrome and what to expect. Genetics and genomics aspects. So, Angelman syndrome is caused basically by mutations to a gene called UBE3A. It's located on chromosome 15. You get one copy from each parent, and it's either turned on or not. Only one copy is turned on in the brain, basically, and that's how you get Angelman syndrome. There are genetic tests you can do through blood work, and genetic counselors often involved. Cytogenetic analysis is one test you can do. That's where they look at the patient's chromosomes and see what arrangements they're in. Also, DNA myelination tests. They basically see which part of the gene is missing from what maternal portion, so father or mother. Hybridization tests, and that can include the FISH or the CGH test. And that's used to determine which portions of the chromosome are missing. Legal and ethical aspects of Angelman syndrome. I find that the newer gene therapies they're trying to come up with could have some legal and ethical aspects. They would work by taking small genetic particles and modifying them to basically go to that certain gene and activate or fix that area, therefore lessening the abnormalities of Angelman syndrome. And they would do that. They have to get past the blood-brain barriers, so they would do that by a vector or a bacteria engineered to be harmless and inject it through your blood, or they would go into your spinal cord intracerebral spinal fluid. There are some possible complications of that, however. Your immune system could react to the vector and go haywire, or you it carries to the wrong spot in your genome and induces disease such as cancer. There are a couple therapies being looked at. One is in a half trial right now, GTX-102, and that's designed to block the expression of the molecule that keeps the paternal copy of the UBE3A gene from being active in the nervous system cells. It is on hold currently because participants in the trial experienced weakness in their legs following high doses, so the FDA shut them down. There is other treatments such as GTAS, and that works by delivering healthy copies of the UBE3A gene to the targeted cells, but those are just being used on animals at this point. There's also another one called CRISPR-Cas9 to try to turn the parental copies of the UBE3A gene on, but again, that's only been done in mice. So all these treatments are in the very early stages and basically years away. But I think, there's a lot of things that could possibly go wrong with trying to do that, but if it were successful and safe, then it could really advance the treatment of Angelman syndrome.